Oh, aku pakai ini. Kalau enggak nanti. Baik, Bapak Ibu, selamat siang. Assalamualaikum warahmatullahi wabarakatuh. Selamat datang di kuliah tamu yang diselenggarakan oleh Departemen Histologi dan Biologi Sel FK-KMK UGM. Kuliah tamu ini kita sangat bersyukur karena dua pembicara yang nanti akan memaparkan materi adalah orang-orang sibuk yang semuanya mau meluangkan waktunya untuk membagikan pengalamannya kepada kita. Sorry, I open this in Bahasa Indonesia first, and then I will try to translate it in English. Dan nanti kita akan ada dua pembicara. Yang pertama adalah James. Nanti saya akan membagikan CV yang singkat. Nanti yang kedua adalah Dr. Gunadi. So James, I will introduce you first to the participant. I hope this is okay for you because I will share. It's a very short CV, very brief CV of you. So ini adalah James McKay, PhD. Beliau adalah Deputy Branch Head Genomic Epidemiologi di International Agency for Research on Cancer. Ini adalah suatu institusi di bawah World Health Organization. Beliau spesialisnya adalah penelitian mengenai genetic susceptibility, terutama pada penyakit pada manusia. Tapi sekarang beliau difokuskan lagi pada kanker yang berhubungan dengan rokok. Penelitiannya banyak sekali, publikasinya banyak sekali. Tidak saya paparkan di sini karena nanti tidak cukup slide-nya. Tapi saya menunjukkan di sini bahwa H-index beliau sangat tinggi. Jadi ini kita bisa melihat dari sini bahwa penelitiannya banyak sekali. So I tell them that you have a lot of research and also publication. So we can see from your citation, your H-index. So I will give the time to you, James, to start this lecture. 45 minutes for presentation and 15 minutes for discussion. So I will stop share to give a chance for you to share your presentation. So time is yours, James.
Terima kasih, Bu Neni. So today I will speak in English. I hope you guys don't mind. I did study Indonesian a long time ago, but as Neni, Yaya, and Dewi know, my Indonesian is not as good, and not good enough to give this presentation in Bahasa. What I would like to do today is give a brief overview of some of our work at the International Agency for Research on Cancer. The plan, I thought, is to give a bit of background about IARC itself, the International Agency for Research on Cancer, and give an idea about our research into cancer prevention. And particularly, as Neni has said, I would like to introduce some of the things that we do with next generation sequencing and more in general, genomics research, and how we use that to understand cancer etiology, and also particularly to understand the etiology of lung cancer as an example. So understanding the biological mechanisms of how lung cancer works, understanding a bit about risk prediction and prevention, and always in the idea of IARC. So what I would like to do is start with what is IARC. I was very happy and very honoured to have both Neni and Yaya come to IARC and work with us for, I think, 2 years and 1 year respectively. And this type of thing, coming to IARC and working and experiencing what it is to be, what the agency actually is exactly what the IARC is for. It's a very old institute. It was actually founded by the famous French general Charles de Gaulle in 1965. And the idea behind the IARC is it's meant to be a centre of collaboration. So that's why we really appreciate and welcome researchers from all around the world, including Indonesia. And we also work to pull together data from many different studies across the world We also do laboratory work. We also host a very large biobank, very similar to what Yaya is developing in Indonesia. And we also do a lot of teaching. We also welcome a summer school and various other teaching facilities. And I would encourage you, the younger students from Indonesia, to look at the agency and have a look at, for example, the summer school and some of the contents, because I think that might be very interesting for you as a learning tool to understand some of this type of research. We are based in Lyon, in France. I am actually from Australia originally, but I have now been here since 2003. So it's been a very long time since I've been here in France. The IARC itself is about 400 researchers, postdocs, postdoctoral fellows, and other students. So it is quite a large institute, but that is a bit of a misrepresentation too, because one of the biggest things about IARC is that while we are 400 in Lyon, we collaborate all around the world. So many people know of IARC and have worked with IARC. And like I said, I'm very pleased to be working with people in Indonesia on various different research projects. So what do we do? So why does IARC, why do we need an international agency for research on cancer? And why this particular mandate to look at cancer research in the service of prevention? Now, IARC itself is affiliated with the World Health Organization, but is also independent. So it's actually funded by 24 different countries from all around the world. That's countries from the West, England, United States, Germany, France, Australia is also a member, but we also have countries like Morocco, Brazil, and Turkey, and Russia, and Qatar, and across Africa. And all of these, the idea behind the agency is really to trying to focus on understudy cancers and cancers that are really relevant to the developing world. And we have three main ideas. First, describing cancer in all its forms across the world. So some of the research that we do is related to the cancer registries. So what we do is we tabulate and quantify the cancer burden across many different countries all across the world. And I know that there is a large initiative in Indonesia to look at developing the cancer registries and having them contribute to the global statistics on the cancer burden. So how and what type of cancers are found in each country. And with that information, we can start to look at how the burden changes, how it's different across different countries. And one clear example that is very relevant, and I work closely with collaborators in Indonesia on, is the example of nasopharyngeal cancer, where in the West, that's a rare cancer. However, in Southeast Asia and in southern China, that is a much more common cancer. And that's the way we try and use these statistics from all over the world to see how cancers change across the world and what we can understand and learn from that. The other side of what we do is understanding the causes of cancer. So this is looking at etiological research. So we can better understand this disease and ideally work on mechanisms to prevent it. And the ideal example of that is something like tobacco research, where we try and understand how tobacco works and how we can ensure that people are able to stop smoking or avoid smoking entirely. But there's also other examples, like looking at viruses and developing vaccines, for example, the HPV vaccine, which is very successful in preventing diseases like cervical cancer. The last action is actually implementing that research to prevent cancer development. And so this is working with local governments all over the world, developing policy to try and help people, for example, to avoid smoking, to implement vaccines or other screening techniques for secondary prevention, looking at early detection of cancers. And those are the three core areas that the IARC works on. However, I work in genetics. So why are we interested in using genetics and genomics to understand cancer? And there's three aspects that I would like to discuss with you today about how we can use genetics and genomics. And this is where next generation sequencing comes in is to try and understand these diseases. Firstly, we can use genetics to identify high-risk individuals. And the classic example of that, and this is something that Nenny, I know, worked on back in the day, is looking at the BRCA1 and BRCA2 breast cancer susceptibility genes. These genes, when they are mutated in a particular form, increase your risk of breast cancer quite markedly. And if we're able to identify people that have those genetic variants, we can then use that to help with their clinical management. Perhaps they can be screened earlier, and we can, or other ways of managing that genetic risk. So there's a lot of work looking at that and trying to extrapolate those types of findings from diseases like breast cancer to rarer forms of cancer. And we could look at examples like kidney cancer. We can look, even now, starting to look at examples like lung cancer in never smokers, and examples like that, where if we can find rare genetic variants or even common genetic variants, we can try and identify those people at risk and try and manage that risk clinically. The second aspect is equally interesting, is that if we can try and identify new genes, we can work on the understanding of the disease itself. So if we're able to identify a particular gene related to either genetic or genomic susceptibility, we can better understand how that disease works. And through that understanding of biology, we might be able to develop better therapeutic targets to work on better drugs to treat these diseases, but also biomarkers to detect these diseases earlier. And also, if we can understand these diseases through the molecular pathways, better ways to understand and prevent the disease from happening from the beginning. And the last aspect, which is again really relevant to what we do at the agency, is that genetics and genomics can also inform us about environmental exposures and gene-environment interactions. Now, this one's a little bit more abstract, so I might just give you an example about how this could work. Now, this is a fictional example. I have not done this in this instance, but can you imagine if we were to find a genetic variant that's related to susceptibility to alcohol or how you metabolize alcohol? If we were to link that variant with susceptibility to breast cancer, that would imply that alcohol has some role in the way breast cancer develops. So by using and understanding the genetics, we can get ideas about how the environment actually works in the carcinogenic process. And we have a great example. The breast cancer example is one I would like to do. I have not done yet, but we have a great example in some cancers of the head and neck, particularly the cancers of the mouth and the larynx, where we see a clear example of where the genetic variants that are related to the disease also are related to how you metabolize alcohol. And this gives you an idea that the alcohol must be related to the causal pathway of this disease. So through the genetics, we were able to establish that alcohol is related to that disease. And so that's how we can use genes and environments together to understand the etiology. And what I would like to do today is talk about one particular disease in lung cancer and understanding, particularly in the way IARC works, understanding the causes of genetic diseases. So using genetics, we can, in the context of IARC, we can looking at individual risk and prognosis, and we can use that in the context of preventing cancer. We can understand if we find new biological pathways, we can understand the causes. And then by using the gene-environment interactions, we can also look at novel environmental exposures. And that's how we, within the agency, use genetics, next-generation sequencing, and other etiological studies to try and understand better and prevent the disease. And so that's our mandate within the genetic epidemiology branch, to use genetics and genomics within the agency. We also, of course, to do these studies, and I think this is something that is really important, is that we also across our branch have a very broad number of individuals that are working in our branch, and we're very lucky in that sense. So we work with very traditional epidemiology, And Yaya is a great example of that. So we're doing large case control studies all across the world. We are also involved in prospective cohorts. So in other words, people that are followed through time to see how they develop disease. And they're extremely important in understanding the etiology. We also have a laboratory work, which is involved in a genetics platform. So we have the next generation sequencing and pathology laboratory. And equally critically, we have the analytical capability. Because one thing that's really important with next generation sequencing and other genomics techniques is that one needs very talented bioinformaticians and biostatisticians and the associated computational capacity, because some of these data sets are very complicated. So we need very powerful computers to be able to analyze this type of data. And across our branch, we're very lucky because our scientists and our research staff have all these different areas, and we're able to work in all these different areas, from field work to managing the samples and also analyzing the data and setting up the right studies to understand the disease. So what I would like to do now is switch to lung cancer. Now, lung cancer is a very important disease for Indonesia. And overall, over the world, it still remains one of the most important causes of mortality in any type of cancer across the world, killing about 1.6 million people each year. And the graph on the right is actually a really interesting one, because here we can see what is the survival by lung cancer stage. And one can see that even though this disease kills many, many people, if you are diagnosed at an early stage, at stage 1, you actually have a pretty good chance of surviving. But the unfortunate part is that most lung cancers are diagnosed at stage 4. So much, much too late for any treatment to work. And unfortunately, that results in a large number of people who die of this disease. So while if we were able to, we could do much, much better in improving lung cancer survival by diagnosing people much earlier. Unfortunately, the burden of this disease is very high because most lung cancers are diagnosed at later stages. So even if we were able to shift this diagram a little bit, say shifting the number of the people on the right that are diagnosed at stage 4 disease, if we could shift them to stage 1 or to even stage 2, we would greatly improve the number of people that are able to survive this disease. Now, lung cancer is, of course, also very much related to tobacco smoking. And I'm going to give you today some examples of how the genetics works and how we've used next generation sequencing to explore this concept of susceptibility. But what I would like you to do is just take away this graph here. If we could just shift these people that are being diagnosed at stage 4 to stage 1, we would do so much better. We would do even better if we can just simply get people to stop smoking. And I know in Indonesia, there is still a large proportion of people that continue to smoke. And I would encourage anyone to try and work with governments and local people, or even just your friends, to get them to stop smoking, because smoking is one of the leading causes of death across the world, and it is preventable. So if we could avoid tobacco smoking, that would be a major, major improvement in public health. Now, what about genetics though? If smoking is all related to lung cancer, why are we interested in genetics? So this graph here is a descriptive graph. What this does is try and quantify the heritability, or in other words, the degree at which genetics is involved with the disease. In this instance, what we're doing is we're looking at what is the risk, the genetic risk in first-degree relatives. Put more simply, if my father or my mother or my sister is afflicted with that disease, what is the risk for me as an individual to develop the same disease? What we can see is, for example, breast cancer or colorectal cancer or prostate cancer. It's about twofold. So if my father has had prostate cancer, I'm about twice the likelihood of developing that same disease. If Nenny's mother has breast cancer, she's about twofold increased risk of developing that same disease. Now, lung cancer is an interesting one because here we see that if my first-degree relative has a lung cancer, we can estimate that there's, again, a doubling of the risk of me developing that same disease. However, one needs to keep in mind that families share more than just their genes. Families grow up together, they are exposed to smoking, they are more likely to smoke together. Therefore, a given person that's in a family that smokes is perhaps more likely to take up smoking. And we can see this demonstrated here, because here this is a different way of estimating heritability using genetics and next generation sequencing. And what this does is uses your genes to try and estimate the heritability of a particular disease. Now that, using one's genes, would be independent to tobacco smoking. So what we can see is that in this instance should be a little bit less biased towards that shared environmental factor. And here what we can see, the heritability for breast, for prostate, is much higher than lung cancer. And that's probably related to that shared early environment where if someone in your family is a smoker, it probably means that later in life, you will become a smoker as well. And that means you have increased chance of dying of lung cancer. Now here, the other thing that's important to see is that not even though it is lower, there is still a good proportion of heritability attributed to lung cancer and genetics. So there are genes involved. We just don't know exactly what they are yet. And we use this technique called a genome-wide association study. This is a very simple technique where we take a bunch of patients and a bunch of apparently healthy individuals or controls. And we simply compare the frequency between these two groups. Is a given genetic variant or genomic variant that has been identified by next generation sequencing, is that more common in patients compared to controls? And as we can see in this example here, the individuals that are green appear to be more common in cases versus those ones in controls. Therefore, this might be related to the disease itself. So that's the very simple idea. And the nice thing is, we're using these genomics techniques. This is not genetic next generation sequencing, although we can be starting to do this using next generation sequencing. But generally, we use these things here, which is another genomic technique called gene B-chip arrays. And these are able to measure all of the genetic variants in a given population. And it only becomes a little bit more complicated because what we do is comparison in the middle here between cases and controls. We do that millions and millions of times. So we need to be very careful with our statistical analysis because we are doing 7 or 10 million comparisons between patients and controls. And as any statistician will tell you, when you do these types of comparisons that number of times, strange things often and will happen. So we need to be very careful about how we do these statistical tests and what we call significant. But nonetheless, it works. This is a graph of the human genome. So this is all the different chromosomes from chromosome 1 on the top left to chromosome, their sex chromosomes on the bottom right. And each little dot is a place in the genome that's been related to a particular disease susceptibility. In fact, there are over 30,000 individual genetic variants that have been linked to various forms of genetic disease. And of those, 3,000 have been related to cancer susceptibility. So there are many, many different genetic variants that have been linked to various diseases, including cancer. And this is through this genome-wide association approach. So we've learned very much about how this works and the genes involved. I was lucky enough to be involved with this one, which is a large-scale study involved in lung cancer. Now, to do these studies, we need very, very large sample sizes. You can see here that we looked at 27,000 cases of lung cancers and 57,000 controls. And we combined 29 different studies from all over the world. from China, from Brazil, from United States, from Canada, all across Europe and Australia. And the crazy thing is we now understand that we probably need another 10-fold increase, in other words, 200,000 cases to do this properly and exhaustively. However, at the moment, we're only at this sample size. Nonetheless, we were quite proud of this study because it also represents an enormous amount of work to put all these data sets together. And that's, again, something that the agency and the way we work with collaboration all across the world is very much involved in. But nonetheless, through these different studies, we found a number of really interesting genes. For example, we found genes involved in DNA repair, such as the breast cancer susceptibility gene, BRCA1, CHEK2, and ATM. This was really interesting because no one previously had thought that those genes would be involved in lung cancer susceptibility, and we were able to show that. Other genes like CDNK2A, which is a melanoma susceptibility gene, and also involved in cervical cancer and P16. Other genes related to telomere length or ends of the chromosomes, really interesting genes like NRG1, and many genes, this is the beauty of this technique, we were able to identify genes that we had no idea were related to lung cancer susceptibility. For example, these at the bottom, RNA-SET2 and SECS-BP2L. We don't know what these genes do, and we're now currently exploring to see how they work. But what I would like to do is really discuss these ones, and these ones are related to propensity to smoke. And so these ones are really related to addiction and how you actually smoke tobacco. And I'll give you a couple of examples of that. So this is one of the first genome-wide association studies that we identified. And this particular locus is on chromosome 15q25. We identified it using the genome-wide association approach. And then when we looked at the genes that are present in this particular locus, we were really intrigued to see the 3 genes on the right, the CHRN genes. Why are they interesting? Well, these genes themselves are called neuronal acetylcholine receptors. And they are involved in how signals in your brain are transmitted from neuron to neuron inside your brain. They are also involved in other biological processes across your body, for example, muscle contraction and so forth, but they have this really interesting effect in the brain themselves. And they are also called nicotine receptors because as you smoke nicotine and it moves through your body in your bloodstream, nicotine will bind antagonistically to these receptors in the brain, resulting in a slight change in the way your neuronal signals are transmitted across your brain. And this is actually how we respond to nicotine. So if an individual smokes, they will inhale that smoke into your lungs. The nicotine is then absorbed into the bloodstream, moves to the brain, and that's where the pleasure from nicotine actually comes from, and also the addiction aspects of it. And what we found is that some of these genetic variants are changed based in terms of their expression and other aspects based on the genotype. You can see in the bottom right, this is the expression levels of a gene called nicotine alpha-5, which is expressed in the brain by genotype. So on the bottom axis, we have the 3 different genotypes, a heterozygote, homozygote common, homozygote rare. And you can see that the expression of this particular gene is correlated with the A allele. If you have one version of the A allele, you have a lower expression. If you have 2 versions of this particular allele, you have even lower expression. And so you can see that these genetic variants seem to influence how these genes function. And accordingly, it is also related to lung cancer. So what we can see here, this is called a forest plot that looks at many different aspects of lung cancer susceptibility. The top is just looking at the genetic variants. The middle one, I see someone is called Histo Lover. I love that name, by the way. And this looks at the different histological subtypes of lung cancer. And we can also look at smoking status. For example, this genetic variant has no effect in never smoking lung cancers. It appears to be only relevant to smoking individuals. And that's consistent with the notion that this effect is mediated through smoking. And indeed, this is what we see. We can see with these individuals, with this genotype, they tend to smoke a little bit more. For example, if you have the alternate variant or the heterozygote variant, you tend to smoke a cigarette more per day compared to individuals that don't. If you have two variants of it, you tend to smoke 2 cigarettes per day more compared to someone who doesn't. That is also true in cases and controls. So basically what this variant seems to do is provoke you to smoke. Now, if you are provoking to smoke, it is more likely that over time you will accumulate somatic mutations. As a result of that accumulation of somatic mutations, that would lead to an increased lifetime risk of developing lung cancer. Lung cancer, we know, is very causally related to tobacco smoking. So anything that provokes you to smoke a little bit more through your lifetime will mean that you have an increased risk of developing lung cancer in your lifetime because of that increased exposure to tobacco smoke. And that's this idea here. And this is how we've used next generation sequencing to try and test this hypothesis that genes will influence your tendency to smoke. This will result in an increased lifetime exposure of carcinogens, and that will result in increased accumulation of somatic mutations. Now, the beautiful thing about next generation sequencing is that we can now use these techniques to work out in a given tissue or a given tumor or even a given bit of normal, histologically normal material, we can actually estimate now the number of mutations found in that tissue using next generation sequencing. And so that's what we did. We tried to estimate the number of mutations. We can even take this a little bit further because there are because of the nature of carcinogens and the way they work, there are even particular changes in our DNA that are related to different exposures. And this is related to the way these carcinogens work. For example, on the bottom left, we see a cigarette. Inside cigarette, there are 150 something carcinogens. And of those, some of them have a particular propensity to bind to the C base. And that's related to the way the carcinogen works. The carcinogen has an affinity, it's a chemical process, it has an affinity for a particular base change. So it will attack Cs a little bit more frequently than Ts. And this is something we call a mutation signature. So for example, in lung cancers, we will tend to see this particular base change, C to A little bit more frequently because of the nature of the carcinogens in tobacco. There are other examples. For example, in skin cancer, which is related to ultraviolet light, there tends to be an over-representation of C to T mutations. Again, because in ultraviolet light has a slight more tendency to attack, again, the C base than other base pairs of DNA. And there are other examples of this, notably aflatoxin, which is found in certain crops, and other aristolic acid, which are found in herbal and traditional medicines. And these all provoke very particular types of mutations. And we can do this a little bit more subtly by looking at the base before and after. So here we're looking at the changes that happen in our DNA and the different base changes, but also the bases before and after. And we can see again that for example, the tobacco mutations, which are the ones at the bottom, tend to have a little bit more CCCs than mutations than other forms of mutation signatures or carcinogens. So what we can do by assaying the tumor we can get an idea of the burden or the amount of mutations that are related to a particular exposure, like tobacco exposure. So what we wanted to do is go through this particular hypothesis. We thought that those with a smoking propensity, in other words, those people that have an increased propensity to smoke because of their genetics, should have more somatic mutations in their tissues. And we tested this using something called a polygenic risk score. So I'm not going to go into this, but this is basically an idea of putting a number of different genetic variants together. So we found many genetic variants related to smoking propensity across the human genome. We grouped them together to divide individuals into those that smoke a lot and have a lot of risk of lung cancer, and those that do not have that genetic propensity to smoke and that don't smoke a lot. And we constructed that into something called a polygenic risk score. And we had about 100 variants to do this. Then, using next generation sequencing and other genomic techniques, we used exome sequencing. So we profiled the tumours of 1,234 lung tumours. We profiled their somatic tumour and also their paired germline. And we also did whole genome sequencing. So the difference here is one technique looks at the coding parts of the genome, and the other one looks at the whole genome, so all the different sequences across the somatic tissues. And then what we did is we tried to see how does the mutation burden, or the number of somatic mutations that are present in the tissues, how does that relate to a polygenic risk score? In other words, how these people are genetically predisposed to smoke. So on the bottom axis here, across the y-axis, there are 5 different quantiles. And this is just simply us dividing up these 1,000 individuals into how genetically related they are to propensity to smoke. So in Q5, those are the individuals that are most likely to smoke and develop lung cancer by their genetic profile. In Q1, these are the individuals that do not have that genetic propensity to smoke. And what we can see is a lovely trend with the mutation load. So what we did is we profiled these tumours, and then we counted how many mutations we see per genome in these patients. And on average, those individuals that have this genetic propensity have many more mutations inside their tumours. And those individuals that do not have this genetic propensity and are able to avoid smoking have many less mutations in their tumours on average. And this is very consistent with our initial hypothesis. We can do the same thing, but then focusing on the mutations that are related to tobacco smoke. So trying to focus on these C to A transitions. And we see the same effect. Those individuals that are predisposed genetically to smoke have more mutations inside their tumours, and those individuals that are not predisposed to smoke have proportionally less. And we also see a trend. So in other words, as you go up across these polygenic risk scores, we see this trend between the number of mutations that we see and the effect on the number of mutations that we observe in the tumours. This is also true with other types of mutations. So this is what we can do with exome sequencing. Genome sequencing. We can have a bit more sensitive because we're assaying so many more bases. We can look at different types of changes. For example, double base pair changes or deletions or many different types of mutations. And again, we see the same effect where the polygenic risk score, which is related to if you are more addicted to nicotine, you also see more mutations inside your tumours. So this provided a really nice proof of principle about how we can use germline genetics to then explore the tumours themselves and how they actually work. So I'm going to finish up. So the idea of genetics and genomics, it can be very informative, particularly when we do things agnostically. When we find things that are unexpected, we can make new discoveries and push ourselves in new directions. We also have all of these biological sets, and this is the amazing thing with next generation sequencing or genetics and genomics, is it gives us so much information. That sometimes can be a challenge, and one needs to be very careful about how one uses this data, but nonetheless it is an amazing resource, the breadth and the depth of information that one gets from these studies, to be able to go into a given person's lung tumour and sequence it like this, and then pull out all of these different somatic mutations to see how this works. Now, what we did show through this lung cancer GWAS is the importance of smoking. Now, you would say, well, James, Richard Dole and Richard Pito, they described the link between lung cancer and smoking in the 1960s. All you did was find the same thing. But I presented this to Richard Pito a few weeks ago, and he was still very happy to see this, because it again shows that we have a mechanistic approach. We can show that by your genetics, which should not be related to anything else because of the principles of Mendelian genetics, that this is causally related to the disease itself. So this really shows that beyond any doubt that smoking is related to lung cancer. And if you smoke, and if you smoke a lot, you will accumulate somatic mutations, which we can observe inside the tumours, and that over your lifetime will result in increased risk of lung tumours. The other thing, though, and this is what I did not talk about today, is that there are many other stories which are equally fascinating. BRCA2, ATM, CHEK2, and all of the ones that we don't know what they do. Each of those have another story, and I could talk for hours and hours and hours about any of those examples if you would like me to. But I thought I'd just give you this idea here today about looking at the propensity to smoke and how that works. So I would like to first thank all of these people, which are the wonderful people that we work with in the agency in Lyon at the moment. And there's this genetic data that we use all sorts of different genetic projects. I'd also like to thank all of these people that contributed to these studies and the funders that helped us do this research. I would also like to thank Nenny for this opportunity to give this talk to you today, and I'd be very happy to give any questions, if there are any. Over to you, Chair.
Okay, so thank you, James, for a very interesting presentation. So now we have a discussion session. At least now there are two questions you can see in a chat box, but I will read it to you. So the first question is from Umar. He asked, actually there are two questions from Umar. The first is, how can it be implemented? I think in this case, next generation sequencing can be implemented within concept from bench to bedside. And the second is how to overcome the challenges in developing biomarker as diagnostic tool for some disease such cancer. Okay, James. So first question first.
Okay, so the bench to bedside is a very important one, because us as scientists should always be focusing on cancer research that matters. There is an argument to do pure academic research. This is because it does push us into unknown territory. And so I would be proponent not to just only focus on what will make it to the bedside, but nonetheless, it is a very important thing to keep on our focus. And for next generation sequencing, this potential that we can translate our findings from our genetics and genomic studies quickly to assist patients across the world is a really meaningful thing. One of the projects that I'm working on at the moment is translating our genetic risk. So if we can triage our genetic risk into to allow us to identify at-risk individuals, one of the most exciting things that has happened in lung cancer field over the last few years is something called low-dose CT scanning. It touches a little bit on the second question too. This technique is able to identify early stage lung tumours using CT scans. And this has been proven through randomized controlled trials to decrease the mortality associated with lung cancer, because we can shift the cases that are diagnosed towards earlier stage, which means we can intervene and these patients tend to do better. The problem with those techniques, and this is very relevant to Indonesia, is that they are very, very expensive. Even in a very wealthy country like the United States, it is simply not possible to offer CT scans to everyone. We need to be very targeted into it to identify individuals that are at high risk, to be able to deploy this technology, because it is simply too expensive per patient. So one of the things from the lung cancer point of view, how to make this work in the context of to the bedside of the patients, from the bench to the lab to the hospital, is to be able to use these genetic profiles to better target those screening techniques. So if we can use a cheaper genetic test, I'm still not saying they're cheap, I said cheaper, but if the CT scan costs $10,000 per patient, if we can do a genetic test that costs $40, if we can identify those at-risk individuals and better target that technology, that becomes much more cost-effective for a particular health system. The other advantage of this too is to be able to better recognise those individuals that are at risk. At the moment, it is simply based on smoking, how much you smoke and for how long. But that doesn't really capture all of the intrinsic things and related to how and what and where you smoke and individual differences. And so maybe the genetics is able to better represent that, and that will allow us to better target these techniques. The other aspect of how we translate these findings into the clinic is the second question, that's using biomarkers. So another work that we do at the agency is looking at biomarkers. My colleague, and I believe a good friend of Neni's, has been working on using next generation sequencing to detect mutations that are related to bladder cancer. This is a very particular mutation that happens in the promoter of the gene called TERT. And the really fascinating thing is that first Florence, my colleague, she looked at a case control series that were diagnosed, the same diagnosed patients, and found that in their urine, you can detect these mutations up to in 90% of cases. And it was very rare in controls. And this is a perfect example of a biomarker. Then she used something even more amazing. What we did is we did it inside a prospective cohort. So these people are people that have given a biological sample, in this instance urine, and then were followed over time. And this time period was up to 10 years. So we had the biological sample at one end, and then we waited over time for these people to develop cancer. And when we did, we were able to then go back to the biological sample and check to see when that mutation was there. And the really fascinating thing using next generation sequencing was that mutation was present up to 10 years before diagnosis. So the people have been walking around with this mutation with no obvious signs of developing their cancer up to 10 years before they actually developed the disease. And so that gives us a big opportunity for intervention. If we can use these types of biomarkers to detect the diseases a little bit earlier, we're able to identify those at-risk individuals and follow them and manage that risk clinically. And that's the promise of various different biomarkers. So there are a number of biomarkers that happen with next generation sequencing, although of late, the most promising ones are actually more simple, that they're actually related to proteins, simply assayed in the blood by a standard ELISA. The only catch is, and these may be coming from the tumor itself, so we can take serum samples or plasma samples and we measure certain proteins in your blood, and they can be used to detect the disease before it happens. And that's a huge advantage, because for any health system, not a rich or poor, it doesn't matter, it is so much more cost effective to avoid a disease for everyone involved. That's in turn cost effective in terms of money, but also pain and suffering and morbidity associated with the disease if we can detect that early. So I see a huge potential for those types of techniques in a place like Indonesia, where if we're able to develop a much more simple blood test that's able to identify people at risk without the complexities of a diagnosis, that would be a major benefit. So that's what I would say for different biomarker work. Now, the biomarkers, we may have to have another talk about that one, because the second question, I could spend maybe 3 hours going through the importance. The most important thing of any biomarker, validation, validation, validation. Now that's in terms of its performance as a biomarker. Can it detect the cancer? Then there's the validation. Is it found in normal people? And you'd be surprised at how often it actually is found in normal people. For example, we did some work on circulating tumor DNA, which is bits of the tumor which is found in your plasma. And we assumed that there's no way you'd find in apparently healthy individuals these DNA changes that are found uniquely in the tumor. But when we looked, and we were one of the first groups that actually did this to look and see and check, everyone had just assumed that they were not found. Hey presto, we did find them in 5% of individuals. So 5% of individuals at age 40 are walking around with what apparently would be a somatic mutation in your blood, which was a really surprising finding and a bit of a problem for biomarkers, because what we need for a biomarker is to very clearly identify those people that have it compared to those that don't. So with biomarkers, one needs to be very careful about study design and trying in biomarker development, working on the best study design to detect and validate a biomarker before we start to use it.
Okay. Thank you, James. Mas Umar, apakah masih ada pertanyaan lanjutan? Atau akan diteruskan, nanti bisa ditulis di chat box lagi. Okay, James, so I will read the second questions. I think it's more about the result of your research. Is it correct that, for example, smoking has greater risk to lung cancer than brain cancer from genomic analysis that had been done before?
Right, thanks. That's a good question. And the answer is yes. But one always needs to remember that smoking is such a strong risk factor for lung cancer. It has effects on many, many other cancers, and remarkably so, unfortunately, at a more modest level. But that's all what you compare it to. For example, with lung cancer, the risk is very, very, very high. So if we compare the risk of lung cancer or brain cancer to lung cancer, then the risk is proportionally lower. But that doesn't mean the risk of brain cancer, isn't it, or other forms of cancer, blood, bladder, head and neck, breast, prostate, leukemia, lymphomas. Smoking is related to all of these diseases. And if you compare the risks of those rarer types of cancers or less traditional cancers compared to people that don't smoke, you still see a very elevated risk. So one thing I would take away is, yes, lung cancer is the most prominent one, but the rest of them are equally as important. One statistic that I find just so sobering is that if you take the mortality associated with tobacco smoking, all forms, because of, yes, tobacco is related to increased cancer risk, but it's also related to increased death through cardiovascular disease, through associated morbidities, COPD, pulmonary problems. The total risks between 1/3 and half of people who smoke that live to the age of 80 will die of that disease. Can you imagine that? That 1 in 2 or 1 in 3 people that smoke will be killed from a disease related to that exposure, smoking. So again, one of the most important things we can do is to stop people to smoke. That's, if I can say anything to you folk on this call, please do that. Terima kasih banyak. Don't stop.
Yeah, thank you, James. We have still one other question, but actually I have one question that probably it's most question of everybody here. That if, for example, we have next generation sequencing and we also have the conventional one, what make we decide which one we choose, NGS or the conventional one?
It depends on what your question is. If you are talking about research or you're talking about the clinic.
Okay, for example, for the research, because yeah, this is in Indonesia. Nowadays we have the next generation sequencing, thanks to Dr. Gunadi. But then if we have, because we the funding most probably not that big, so when then we decide, but our research question most probably need to do in NGS, but the funding is only possible for the conventional one. So what should we do then?
Absolutely. It depends. Again, I think for a country like Indonesia, huge country, 160 million people, and even then, like for example, lung cancer. In Indonesia, I know the smoking habits are very different. The clove cigarettes, this is understudied. What is the impact of clove cigarettes on the somatic mutation burden in lung tissue in Indonesia? Simple question. I don't know the answer to that. I would love to be able to do that type of study, to look at Indonesia and the particularities of your country and see how that relates to the cancer burden. And I think you can do that quite cost effectively by choosing the right samples. So one thing that I think, I don't know if she's still on the call, but Yaya is doing, and I would encourage this, is to collect the samples. That's perhaps what I would do first, because next generation sequencing, some of the things that, as you know, Neni, the samples that you need tend to be a little bit more tricky to be processed.
Yeah.
So it's more difficult to do next generation sequencing on samples that have been fixed in formalin, for example, or other things like that. So if we can use collect the samples and make sure that they're frozen rather than kept in a -80 freezer, rather than fixed in formalin, formalin's still possible, then that opens the opportunities for next generation sequencing. And to make that, because I totally agree, they are very expensive techniques. Whole genome sequencing is getting a lot cheaper. It's now under $1,000 per person to do a whole genome, but still, that's $1,000 per person. There's an interesting machine that is being developed in California at the moment that promises to decrease that to $100 per person. Now that means that the data that is generated will be more expensive to store, to analyze, than the actual sequencing itself. So don't forget the bioinformatics at the other end. There is a hidden cost there. So when we say, oh, this costs $100, yeah, but it costs $200 to store it. So we need to also remember that there's the other side. But the most important thing is these technologies change, become more cheaper to be used, that Indonesia, through your collection of the samples now, has the opportunities to then study them over time. And I think what I really think is fascinating, and I talk to Yaya about this, is her work developing this biobank. And by that, I think that you then, when you have those right samples, you'll be able to exploit them, the results.
Okay, thank you, James. We have another 2 questions actually. This one I try to, because it's very long, I try to summarize it, and it seems that NGS doesn't have a standard modality for detecting genetic disorder, and there are no genes group panel yet for bioinformatics, and number of genes covered in panels affect the number of things. The more target genes, the less coverage is produced, and okay, to produce wide coverage required cost is greater. Okay, so is there any solution for the problem above?
Not an easy one. I mean, I think the ideal solution is that we move to a cost of about $100 per genome. And so therefore we get reasonable coverage across the genome. And this is a problem that is being assessed by a number of different healthcare systems right now. How do we make this more cost effective? The United Kingdom, England, is doing a large study on 100,000 individuals that have been sequenced by whole genome. And they are now doing a cost-effectiveness study to work on the best way to do this. Is it better to sequence a targeted panel for a given disease, or is it better to do a whole genome and then have that profile for that individual and use that over time? These studies are underway. I know the French are also doing a particular study. And I think from a setting like Indonesia, I think we can learn a lot from those types of studies to how to cost effectively implement these programs. The other thing that is very interesting at the moment is there are a number of companies. For example, there's one called, I shouldn't say what the companies are, it's not my job, but there are a number of companies that are now developing these profiles and also the bioinformatics to help, for example, clinicians assist people in their management of the data that happens from it, also pathology labs and so forth, to assist like in any health system or any clinical test. There is now, as we learn and understand next generation sequencing more and more, this is being implemented by more and more clinics and therefore becoming a little bit more simpler. So I think, for example, targeted panels are now clinically used in most in most hospitals now, but they're still based on targeted panels, and they are searching for genes like KRAS, like EGFR to modulate the drugs of choice for those particular patients. That's in clinical practice here in France at the moment and across Europe and the United States. What I would hope is that we use these genomic technologies not only to assist in treatment choice, but also for early detection. And I'm actually also really interested in the proteins because they are so much more cost effective, because next generation sequencing, amazing research tool, but as a health system tool, the cost is still very high per patient. And if we can make that a bit more cost effective, particularly for lower middle income countries, I think that's amazing. And what I'm excited to see is that the performance of the proteins is as good as the next generation sequencing. So if we have the choice, why not spend 40 euros on an ELISA than 400 euros on sequencing? I think that's a really exciting opportunity.
Yeah. Okay, James, thank you. So
just one last point. That does not mean that the next generation sequencing isn't an amazing research tool, because I just gave the example of lung cancer. But in Indonesia, I also know that there are chewing tobacco, there are other things that happen in Indonesia which are really specific to your health system. And also nasopharyngeal cancer, what I would like to work with with Yaya on. How does that work in Indonesia? And I'm very interested to also hear the talk later today about your work in COVID and looking at the variants in COVID. And okay, there's one last question and I should pass it over, I think, to our speaker.
Okay, I think we have like 2 minutes. Yeah, so based on your study, how about the roles of glutathione reductase genes, which could affect the risk of lung cancer caused by air pollution?
Sure, and that's an interesting one. I mean, the majority of lung cancers that we see are tobacco related, and these types of genes, the metabolizing genes, they are of relevance. One can be very mindful of how this might work. Is if you inhale any given carcinogen from tobacco smoke or from particulate matter from air pollution or things like that, if you have a genetic difference about how you metabolize that carcinogen, that could lead to a change in how your DNA is attacked and repaired. And indeed, this is what we see. The reductase genes, not so much, but particularly the CYP1A1, we've just identified it's a new finding which is related to the metabolism of these particular carcinogens. Yes, we have related those genes to risk. The risk is only modest, but nonetheless it is there. Yeah. So the difference between metabolism is only minor, but if you have it over your lifetime, then it starts to manifest as a difference in genetic risk.
Okay, So everybody say thank you. I also thank you a lot, James, for your time. We give a virtual applause to you.
Thank you very much.
And I think before we move to the next speaker, I would like to ask everybody. Oh, yeah, it's raised hand. Okay. Yeah.
No, it's a mistake. Please
it's a mistake.
Yeah.
Okay. Okay, so I would like to ask all the participants to open your camera. So we will have a photo session, please. Okay, everybody's inviting to open the camera. So James, you can see everybody here. Okay, yeah. Okay.
And thank you very much. Terima kasih banyak. Terima kasih banyak.
Oke, Mas Satrio mungkin bisa mulai menghitung untuk
Baik, Bapak Ibu, ada 2 layar. Saya tangkap layar pertama, ditahan streamnya 1, 2, 3. Layar kedua. Ya, sampun Bu.
Matur nuwun. Okay, so we are moving now to the second presenter. I will use bahasa Indonesia, James. And if you would like to follow, you may, because the slides in English. So I hope that you can understand. Bapak Ibu, berikutnya presentasi berikutnya adalah oleh Dr. Gunadi. Saya akan mencoba untuk menayangkan. Oh, maaf sebentar. Ini tadi maaf. Ini mengapa saya sebentar, saya mau menayangkan CV-nya Dr. Gunadi. Kenapa yang keluar yang lain ya? Sebentar. Mungkin tidak mau ditayangkan ya CV-nya ya. Oke, saya membuat CV singkat karena tadi saya mendapatkan CV-nya panjang sekali. Jadi Dr. Gunadi ini S1 dari Fakultas Kedokteran, S3 dari Kobe, kemudian melanjutkan dengan Research Fellow di Johns Hopkins, kemudian mendapatkan kesempatan banyak sekali sampai dengan tahun 2020. Sampai saat ini Dr. Gunadi juga bertindak dengan banyak sekali, ini sebetulnya panjang, saya hanya ambil yang dari tahun 2021 sampai 2020. Mohon maaf ini Dr. Gunadi, saya singkat. Tapi yang paling baru ini adalah sebagai Presiden untuk Indonesian Society of Human Genetics, juga berperan serta sebagai lead guest editor untuk beberapa macam artikel. Kemudian kalau kita lihat juga ini ada banyak sekali penelitian yang dilakukan oleh Dr. Gunadi. Saya juga ambilnya dari tahun 2002 sampai 2020, karena kalau dilihat dari itu nanti mungkin akan banyak sekali. Beliau juga sangat rajin untuk melakukan publikasi, sehingga tadi bisa dilihat high indeksnya sangat tinggi, dan sekitar kalau tidak salah ada 77 publikasi dari Dr. Gunadi. Baik, saya akan stop share untuk memberi kesempatan kepada Dr. Gunadi untuk memberikan penjelasan mengenai bagaimana peluang dan tantangan untuk melakukan next generation sequencing di Indonesia. Waktunya 45 menit, Dr. Gunadi, waktu saya serahkan. Monggo.
Bu Neni, Assalamualaikum warahmatullahi wabarakatuh. Sorry James, I will present in Bahasa, and I do hope that we can, actually I learned a lot from your presentation. Then I will present the challenges in Indonesia that you can give us suggestion for the problem facing in Indonesia regarding NGS research. Bapak Ibu yang terhormat, yang kami banggakan dalam kesempatan ini, terima kasih kepada panitia Bu Neni dan Departemen Histo yang telah memberi kesempatan kepada kami untuk sharing pengalaman kami, peluang dan tantangan penelitian NGS di Indonesia. Jadi ini tadi 550 since November 2021, and soon to be Great Iron in Dr. Sarjito Hospital funded by Ministry of Health. Bapak Ibu, mungkin tadi juga sudah disinggung beberapa oleh pembicara sebelumnya bahwa aplikasi dari sequencing itu, ini saya ambilkan dari slide Illumina, tergantung kita mau mengambil penelitian DNA, RNA, protein, dan sebagainya. Banyak sekali yang mungkin penelitian kita kan sangat beragam ya. Alhamdulillah ini Bu Neni pesertanya cukup lumayan banyak ya, lebih dari 60 dari awal sampai akhir. Sehingga Bapak Ibu harapannya mungkin nanti penelitian NGS di Indonesia semakin membaik dan tentunya bermanfaat tidak hanya untuk riset tetapi juga untuk pelayanan kepada pasien. Mungkin ini pengalaman yang kami sharing adalah yang telah kami lakukan dan akan kami lakukan ke depannya. Yang pertama adalah tentunya complex genetic disorder, ini yang kami tekuni saat ini, Hirschsprung disease, yaitu kelainan functional obstruction. Yang menariknya adalah insidensi di Asia lebih tinggi daripada Eropa, dan ini jelas berhubungan dengan genetic background dari populasi Asia. Laki-laki juga lebih tinggi. Karena banyak dia multifaktorial, maka tentunya peran NGS di sini sangat tinggi, sangat breakthrough dibandingkan kalau dulu mungkin dengan Sanger kita harus sequencing satu persatu gene, tetapi dengan NGS maka sangat satu kali eksperimen bisa dapat langsung exome sequencing misalnya. Kenapa menjadi menarik? Karena penelitian Hirschsprung itu kita dulu sebelum pre-GWAS kita masih mengenal ada linkage analysis, candidate gene analysis, kemudian dengan GWAS pendekatan common variant, antara mungkin tadi kalau lung cancer juga lebih waktu GWAS juga post GWAS dengan teknologi next generation sequencing. Nah ini tadi sudah disampaikan ternyata waktu sebelum penemuan NGS itu masih sangat minimal 20% ya. Kira-kira pasien Hirschsprung bisa ditemukan mutasinya. Tetapi dengan harapannya dengan NGS ini maka bisa meningkat lebih dari 20%. Nah ini pengalaman kami yang telah kami lakukan. Kami melakukan whole exome sequencing pada 20 pasien Hirschsprung. Tetapi ini sudah meningkat sekitar 39 karena 2 kali running dan kontrolnya 4 kontrol. Tetapi juga kita melibatkan kontrol pasien-pasien ini di sini kalau tidak salah ada Dr. Agung, pasien-pasien neurogenetics. Jadi kita switch. So James, we switch control between our disease and another disease. So we have running together with the patient, then and we compare each other for as control, vice versa. Dan tentunya setelah melakukan exome sequencing kita melakukan bioinformatik analisis dan kita menentukan allele frekuensinya di bawah 1%. Dan kita bandingkan dengan in silico prediction tools, ada banyak beberapa, salah satunya PhyloP, CAT score, Grantham score, dan sebagainya. Kemudian kalau untuk menentukan patogenik, tentunya kita menggunakan SCMG classification tahun 2015. Jadi kita menggunakan istilah varian dibandingkan mutation atau polymorphisme. Ada beberapa istilah yaitu patogenik, likely pathogenic, uncertain significance, likely benign, ataupun benign. Nah kemudian thanks to Dr. Galuh, diaspora dari Undip yang ada di Redboard, karena tentunya ini salah satu tantangan kami dalam bioinformatics. We have problem to perform bioinformatics, James. So we have collaboration with Dr. Galuh, diaspora from Universitas Diponegoro in Redboard University Medical Center. Dari 39 pasien itu didapatkan sekitar 2.000 lebih dari 2 juta varian. Kemudian yang unik varian sekitar 1,5. Kemudian kita filter yang pertama, yang sinonimus kita buang sehingga kita hanya memasukkan amino. Dan kemudian minor allele frekuensi dari 3 database, Genome AD, ESP, dan Exact, sehingga didapatkan 42.000 dari 2 juta sekian awal. Nah kemudian kita filter lagi, hanya kita fokus pada non-coding region. Ya, meskipun exome sequencing, tetapi tetap ada sebagian kecil yang mungkin dari bukan non-coding. Sehingga kalau kita menggunakan strict kriteria, maka ada 444. Kalau yang broad kriteria, maka kita mendapatkan sekitar 2.900. Kemudian kita strict lagi menjadi 1 dan 72. Apa yang dimaksud strict kriteria adalah misalnya stop codon. Jadi perubahan asam amino menyebabkan TAG, TGA, satunya saya lupa, maaf. Kemudian atau splice site yang canonical, yaitu baik itu akseptor maupun donor splice site, atau frame shift, indels. Kemudian yang broad itu apa? Ya misalnya splice region tetapi bukan lagi akseptor tapi donor, lebih proksimal lagi atau lebih distal lagi. Dan kemudian missense juga dimasukkan seperti itu. Kemudian kita menentukan, Dr. Galuh menentukan bahwa disebutkan damaging jika ada 3 tools yang tadi disampaikan di depan itu semua damaging terhadap protein fungsinya. Nah kemudian setelah itu kami mendapatkan beberapa, ini sudah kami sedang under review di jurnal. Jadi ditemu novel gene, salah satunya adalah ini faktor risiko untuk CRC, colorectal cancer. Dan kemudian tentunya ada novel varian, artinya gene-genenya sudah pernah dilaporkan tetapi variannya belum pernah. Ataupun yang sudah established, baik gene maupun variannya. Itulah salah satu kelebihan exome atau NGS. Yang established gene seperti RET, NRG1, SOX10 itu sudah established gene. Homozygous mutation, kemudian compound heterozygous. Jadi dalam satu gene 2 mutasi. Dari epigenetics, yaitu di luar dari fungsi gen itu, misalnya metilasi dan sebagainya, histone modifikasi. Kalau kita melihat komposisi korelasi dari peran bagaimana gene expression epigenetik DNA dan phenotype, itu sebetulnya sangat erat hubungannya. Tidak bisa kita berkata bahwa hanya fungsi gen yang berperan pada suatu disease phenotype, tapi lebih ke arah sekarang khususnya yang complex genetic disorder ada peran dari epigenetics di situ. Kemudian kami mencoba melakukan RNA-seq transkriptom pada sampel kami. Ini juga ada sebetulnya memang ada websitenya ya kita untuk menentukan kita RNA mana yang berperan seperti itu. Nah ini sama ya kira-kiranya. Nah ini beberapa sampel kita fokus pada microRNA. Tentunya karena tadi sebetulnya kita juga bisa menangkap RNA-nya, microRNA. Mungkin sekarang banyak juga non-coding RNA yang lain seperti circular RNA, long non-coding RNA, tergantung bagaimana kita ingin fokus kemana. Dan ini juga menggunakan NGS. Dan pengalaman kami sejak tahun 2020, kami menggunakan mesin MySeq untuk COVID-19 whole genome sequencing SARS-CoV-2. Ini beberapa hasil penelitian kami yang sudah dipublikasi dan sedang under review di salah satu jurnal. Ini salah satu peluang Bapak Ibu, jadi kalau kita bisa menangkap peluang bahwa dalam suatu blessing in disguise ya, kita mungkin tidak menyangka ada pandemi, ternyata dalam segi riset itu juga mendorong penggunaan NGS luar biasa, termasuk pengadaan mesin NGS oleh Pak Menteri Kesehatan itu karena pandemi ini juga. Seperti itu, dan kemudian hepatitis. Ini kemarin yang sempat rame, James, we have also involved in hepatitis unknown etiologies, and we have collaboration with Nusantik in Jakarta, and we run 2 sampel from Sarjito and 6 from Jakarta. Salah satu sampel dari Sarjito itu menariknya memang ini metagenomik Bapak Ibu, jadi shotgun metagenomik itu rumitnya lebih rumit, lebih kompleks daripada exome ataupun transkriptom, apalagi SARS-CoV-2 ya. SARS-CoV-2 itu kalau sekali genome sequencing ketemu ini variannya mudah lah. Tapi begitu kita bicara shotgun metagenomik itu yang berhasil kita amplifikasi adalah tidak hanya mikrobiom yang ada di dalam sampel tersebut, tetapi juga genome dari hostnya. Dalam ini kita manusia. Tapi kemudian dengan teknik tertentu maka kita akan mendapatkan yang sampel dari mikrobiomnya, sequencingnya. Tetapi perbandingannya sebetulnya lebih banyak hostnya, mungkin 90% lebih itu adalah miliknya host, sehingga ini untuk metagenomik itu agak sedikit kompleks analisisnya. Dan kami pun banyak belajar dari Nusantik bagaimana dan klinisi tentunya, menentukan klinisnya. Misalnya seperti kasus pertama ini, ditemukan ternyata CMV positif. Tapi waktu kita mengatakan ini CMV penyebabnya hepatitis anon-etiologis tadi. Masih banyak yang perlu didiskusikan dengan klinisinya dan masih banyak ada beberapa hipotesis. Karena selain viral DNA yang dideteksi, bakterial DNA juga bisa dideteksi dari serum pasien tersebut. Seperti itu. Pada kasus 2 juga mirip, lebih yang yes adalah sama-sama herpes viridae, tetapi di sini ada Epstein-Barr virus yang bisa dideteksi. Bakterial DNA-nya juga tentunya komposisinya cukup berbeda ya. Sehingga dari 2 kasus ini dibandingkan dengan kontrol tentunya, kita juga dalam 8 sampel ditambah 1 kontrol, waktu itu running sekitar bulan lalu. Hipotesisnya apakah yang pertama herpes virus tadi ya, apakah CMV maupun Epstein-Barr virus tadi sebagai menyebabkan immunosuppression pada pasien yang hepatitis anak tadi, yang menyebabkan sepsis sehingga akibatnya hepatic failure. Ini hipotesis pertama. Kemudian yang kedua, hipotesis kedua kebalikannya, meningkatnya amplikon dari herpes viridae baik itu CMV maupun Epstein-Barr virus tadi disebabkan oleh sepsis bakterinya tadi, menyebabkan immunosuppression. Jadi ini sangat menarik analisis. Mungkin nanti James you may have comment on this hepatitis anon-etiologis ya, whether hypothesis 1 is which one the more appropriate hypothesis for our case. And then ini yang mungkin akan soon to be ya. Jadi program bio ini Mbak Yaya yang terlibat di pusat sebagai tim biobanknya. Kemudian ini masing-masing rumah sakit ada 6 genomic hub yang ditunjuk, salah satunya Sarjito untuk genetic disorder. BGSI Biomedical Genome Science Based Biomedical Genome Based Science Initiative. Nah ini SK-nya Sarjito ditunjuk sebagai genomic hub untuk genetic disorder. Kemudian untuk saat ini memang fokus pada 2 genetic disorder. Dalam hal ini Mendelian diwakili oleh Duchenne muscular dystrophy, sedangkan kompleks genetic disorder diwakili oleh pulmonary arterial hypertension. Nah, kami juga sudah berdiskusi dengan tim BGSI dan juga tim ONT bahwa karena kita akan mendapatkan Great Iron Sarjito, di mana platformnya ONT, Oxford Nanopore Technology, sedangkan yang sudah kita punya adalah tadi Illumina, yaitu Nexus 550. Sehingga yang kita sepakati adalah akan menggunakan targeted panel sequencing, bukan exome sequencing untuk DMD maupun PAH. Ini adalah bagaimana kita alurnya untuk pasien-pasien DMD sebelum dilakukan pemeriksaan NGS. Tentunya MLPA terlebih dahulu sekitar 60%, baru sisanya yang tidak bisa dengan MLPA karena jenis mutasi missense misalnya atau stop codon misalnya, maka ini memerlukan NGS. Pertanyaannya adalah waktu itu yang didiskusikan oleh dengan tim BGSI, apakah kita mau exome ataukah mau targeted panel? Karena di BGSI pusat itu akan dilakukan whole genome untuk sampel yang sama. Sehingga akhirnya disepakati bahwa di masing-masing genomic hub akan dilakukan targeted panel sequencing. Karena kita kalau melakukan exome semacam redundant ya, karena juga akan dilakukan whole genome oleh pusat untuk masing-masing sampel. Kemudian ini analisis tesiernya waktu itu masih berdasarkan exome sequencing, tapi tentunya ini tinggal yang West itu diganti dengan targeted panel sequencing. Mungkin kalau di MD sekitar 116 gen. Tadi salah satu bertanya ya berapa gen itu. Ini memang kita diskusikan kemarin bahwa betul kalau makin banyak gennya tentunya coveragenya akan turun. Terus kalau itu maka memang itu menjadi seninya di situ NGS itu. Bagaimana kita kiak-kiuk kata orang Jogja itu menghasilkan hasil yang valid dan reliable. Kemudian ini yang akhirnya panel disepakati, salah satunya yang kita ambil dari paper sebelumnya menggunakan targeted panel sequencing, dan menariknya dengan ONT itu we have interesting discussion with ONT yesterday, James, that for ONT platform we do not need to design the primer for targeted panel sequencing. We just upload the sequence of targeted genes into the program, and then if the flow cell during the NGS detect the genes that already uploaded as a sequence in the machine, then it will appear the result. Yeah, it's very interesting. And then ini yang PAH di Sarjito, timnya Dr. Wulan dan kawan-kawan ya. Ini juga kami kerjasama dengan Dr. Galuh untuk menentukan ini apa namanya tertiary analisisnya. Yang salah satu yang mungkin perlu dipikirkan oleh Bapak Ibu bahwa genome sequencing tentu makin rumit tertiary analysis dibandingkan exome. Exome sequencing lebih rumit daripada targeted panel sequencing, sehingga itu hal-hal yang mungkin perlu diperhatikan sebelum kita memilih kira-kira kita itu NGS mau pakai yang mana. Nah ini mungkin meskipun di sini dikatakan panel 23, tapi dari publikasi yang ada itu baru ada 13 gen untuk PAH yang mungkin akan dimasukkan di dalam targeted panel sequencing-nya. Meskipun tadi dengan pendekatan adaptif ya, mereka menyebutnya adaptif targeted panel sequencing, itu tidak perlu lagi mendesain dari primernya. Cukup kita targeted gen yang mau ditarget dimasukkan sequencingnya. Itu akan begitu di running di mesin, maka begitu mendeteksi ada mutasi varian di dalam gen itu, maka akan keluar hasilnya. Seperti itu kira-kira. Kesimpulannya mungkin NGS itu merupakan peluang yang sangat besar bagi peneliti di Indonesia, karena sekarang banyak institusi yang akan mendapatkan mesin oleh Menteri Kesehatan kita. Kita harus berterima kasih kepada Pak BGS, Budi Gunadi Sadikin. Ya mirip-mirip lah namanya, saya Gunadi, beliau Budi Gunadi Sadikin, cuma beda nasib. Kemudian untungnya lagi beliau tidak hanya memberikan kepada rumah sakit vertikal, tetapi juga memberikan kepada Fakultas Kedokteran. Yang kedua adalah aplikasi NGS tergantung dari tujuan penelitian kita. Tentunya kita tidak semua menyelesaikan masalah dengan genome sequencing, semua masalah akan terselesaikan. Karena tadi ternyata tertiary analisisnya, bioinformatiknya sangat rumit dibandingkan exome dibandingkan. Tentunya masalah the most challenges in NGS research itu so far adalah bioinformatics. Kalau wet lab-nya Bapak Ibu yang sudah terbiasa pipetting itu tidak akan kesulitan ya. Cuma mungkin memang dibutuhkan presisi, meticulous. Mungkin kalau kita operasi seperti saya, ahli bedah anak, itu bedah anak membutuhkan meticulous prosedur. NGS sama, meticulous prosedur. Dia tidak bisa sembrono seperti PCR biasa, karena sangat perbedaan DNA sangat kecil itu bisa menyebabkan hasil yang berantakan. Itu mungkin sudah bedanya. Jadi artinya pada dasarnya NGS membutuhkan presisi yang sangat tinggi dibandingkan mungkin PCR biasa atau katakanlah ELISA atau yang lainnya. Itu menurut pengalaman kami, jadi itu yang bisa kami sharing. Mudah-mudahan nanti harapannya bisa mendapat masukan dari James dan Bapak Ibu semua. Terima kasih Bu Neni. Ini terima kasih tim kami sangat luar biasa yang telah sampai sejauh ini ya, sejak 2017 akhirnya bisa goal, alhamdulillah ya. Bu Dewi juga ikut mengamal waktu itu sebagai ketua LRT, mengawal naskah akademik bersama-sama dengan Pokja Genetik, sehingga dari 2017 alhamdulillah gara-gara pandemik juga ini James, we got the machine because of pandemic, blessing in disguise. We have already applied for the machines to UGM since 2017, but finally we got at November 2021 due to pandemic. Terima kasih Bapak Ibu. Assalamualaikum warahmatullahi wabarakatuh.
Terima kasih Dr. Gunadi. Monggo kalau Bapak Ibu ada pertanyaan atau mungkin begini, mungkin saya akan menambahkan tadi juga digarisbawahi oleh James mengenai tantangannya ya untuk NGS itu. Yang pertama tadi selain bioinformatics-nya, tadi Dr. Gunadi menyebutkan juga dengan caranya yang harus memang membutuhkan orang yang skillful ya. Jadi memang perlu orang yang sudah biasa di lab. Kalau orang yang baru di lab terjun tiba-tiba mengerjakan NGS juga tidak bisa, karena terus terang kita akan bekerja dengan volume yang sangat kecil, yang mungkin kalau kita orang awam itu tidak kelihatan, ini sih kelihatan. Lalu yang lainnya adalah mengenai tadi James juga mengatakan mengenai sampelnya. Sampelnya sendiri itu juga harus sangat integrasinya harus baik, karena kalau tidak itu nanti akan mengganggu proses selanjutnya, seperti yang pernah saya alami juga, atau tadi James juga mengatakan terutama kalau itu dari jaringan, itu tidak disarankan sebetulnya dari parafin, karena parafin itu tingkat kesulitannya tinggi dan prioritasnya kemurniannya juga tidak terlalu bagus, sehingga kalau untuk dilakukan next generation sequencing itu agak sulit. Baik, ini James sudah raise hand. Okay, James, I give an opportunity to you to discuss with Dr. Gunadi.
Thank
I
you.
hope no question, just suggestion, James.
No, of course I do. First thing I would like to say is congratulations, because setting up a next generation sequencing laboratory is technically very difficult, and then also to do the bioinformatics, as you correctly pointed out, is a formidable challenge. And I congratulate you for setting this up and doing the interesting work that you've done today, and also to be the head of the Indonesian Human Genetics Society. That's fantastic. I'm very pleased to meet you in that context as well, because that's also very close to my heart. And please do reach out. Both Neni and Yaya have my contact details. If there's any way we can assist you, please let me know. I thought your work was really interesting. The Hirschsprung disease is really interesting. I have a number of questions for you. None of them nasty, don't worry. I would also make this comment, and this is one of the biggest challenges that we have when we're looking at very rare genetic variants, the ones that are usually related, is looking at what's actually found in a given population. And so one of the bigger challenges we have, for example, we're doing some studies in Africa, is what is just prevalent in the populations. When we see these rare, very rare genetic variants, sometimes they can be common in an unusual population or understudied population, because so much of the sequencing has been done in Western countries at the moment. And one of the biggest problems, I think, or challenges that you guys have is using that background populations, like the various different databases are so biased towards Western populations, and having a reasonable panel of individuals sequenced, normal people, just apparently normal, no one's normal, but apparently normal people sequenced to allow you to triage the variants that you find in Indonesia. And even within Indonesia, it is so heterogeneous. Sumatra is so different to Timor. So the variants that you find will be very specific to those different regions. So having a population database of these people to sequence would help enormously to triage the variants. But nonetheless, I thought the finding with MUTYH, the colorectal cancer with Hirschsprung's, you said that's in submitted that paper? That seems to be a really interesting finding. And also the viruses, I think again that is a massively understudied area, how the viruses are present in Indonesia, how they change across different places, and even the different strains in Indonesia. That's something that's really of interest at the moment. For example, in EBV, looking at the different strains across Indonesia, and just those simple descriptive studies would be immensely valuable for the research community. So thank you very much, and I'm really pleased. And please do not hesitate to reach out to me if you need some help with the bioinformatics or things like that, because we all go through that setup project, right? That setup part of trying to get a pipeline in place to be able to triage those variants. We do it at the IARC, the United States labs do it. We've learned a lot over the time, and if we can help you in any of our learning processes, please let us know, because it can be challenged. And also the GridION, the MinION platforms, they have their own particularities as well. but also some great advantage, particularly for viruses, because you can sequence the whole genome in one go. Very long range sequencing. So congratulations. It's really great work, as well as being a clinician too. So as we say in French, je tip de chapeau. I give you a tip of the hat. Well done.
May I comment to
Yes.
Dr. James? Yes, thank you, Dr. James, for your suggestion regarding the control genomes. Yes, from BGSI program, we also the BGSI center will also perform the genome sequencing, whole genome sequencing of the control population as well. So our sample, the control, and also the patient will be sent to the center Jakarta to undergo whole genome sequencing, and then for each genomic hub we'll perform targeted panel sequencing. Then we'll the data and how to say the deal between a genome BGSI center and each genomic hub is only each genomic hub can analyze the data whole genome sequencing. The BGSI center will not analyze the data. That's the, maybe we can discuss with you how to analyze such big data, whole genome sequencing of control and also patients. At least for our genomic hub, DMD and PAH. Yeah, thank you.
And that hub is in Jakarta?
In Jakarta, actually there are six genomic hub. For Cipto Mangunkusumo will perform diabetes mellitus MODI, and then Sanglah will perform psoriasis. Sulianti Suroso Hospital will be perform genome sequencing of tuberculosis. And then Rumah Sakit PON, Otak Brain Hospital in Jakarta will perform stroke, Claudi Progrel sensitivity, and then Dharma East Cancer Center Hospital will perform breast and CRC, and then all sample will send to BGSI Center in Ekman Institute. It will be at maybe you heard about Ekman Institute. Then the BGSI Center might be in the Ekman Institute. Then they will perform whole genome sequencing for each genomic hub in the 6 hospital. And each genomic hub only perform targeted panel sequencing, not exome sequencing. I think we will have big data, very big data, including, as your suggestion, the control, genome control. Very interesting data. Big data from Indonesia will be soon.
Great.
Yeah.
Okay.
It's our Ministry of Health.
Okay, so I hope in the future you will have a more collaboration. I think James already offer and help from IR. So I think Dr. Gunadi can follow up this event. Okay, kita akan membahas ada 2 pertanyaan Dr. Gunadi. Yang pertama dari Dr. Paranita Veronica. Pertanyaannya adalah, apakah selama ini hanya menggunakan fresh tissue atau pernah menggunakan blok parafin untuk next generation sequencing? Hasilnya bagaimana?
Ya, yang pernah kita kerjakan belum pernah terus terang dengan blok parafin, baru dengan fresh tissue dan blood atau serum. Yang terakhir untuk hepatitis unknown etiology itu serum. Kemudian kalau yang Hisprung itu dengan fresh tissue kolon, spesimen kolon langsung fresh tissue. Kemudian kalau yang VTM ya SARS-CoV-2 ya. SARS-CoV-2 itu VTM, kemudian nanti yang DMD PAH itu kita rutin rencananya dengan darah. Sebetulnya DMD sudah rutin juga dengan darah sebelum kerja sama dengan BGSI itu programnya kan kita sudah rutin melakukan exome sequencing juga untuk DMD. Seperti itu. Kalau pengalaman kami dengan PA itu baru PCR FRLP ya, yang memang mungkin kualitasnya memang kalau dibandingkan flash tissue atau blood kurang ya. Mungkin ya secara tadi sudah dijelaskan oleh Bu Neni, oleh James ya, kalau parafin sepertinya tidak dianjurkan. Tetapi kalau mau nyoba boleh-boleh saja, tetapi perlu diingat cost-nya kan sangat mahal ya. Kita mungkin akan rugi flow cell-nya itu yang sangat mahal. Dokter Veronica kan juga melakukan NGS waktu di Belanda. Jadi artinya tahulah berapa cost-nya. Kalau exome sequencing dirupiahkan itu kan satu sampel sekitar 15 juta. Nah, apakah kita mau sacrifice 15 juta untuk satu sampel parafin block? Itu kan tentu kita berpikir ulang. Seperti itu mungkin Dokter Vero. Kalau pengalaman kami yang sudah rutin mengerjakan di Indonesia, satu sampel itu kira-kira 15 juta exome sequencing. Tentunya itu perlu 1000 kali berpikir ya. Kalau itu gagal, tentunya kita bisa menangis Bombay katanya begitu. Mungkin begitu Bu Neni, Dokter Vero. Terima kasih pertanyaannya.
Ya, mungkin sedikit berbagi. Dulu teman saya postdoc waktu di IARC itu, James, probably you still remember Felipe? I think Felipe doing the NGS on paraffin block tissue. Do you have any idea what happened next? Mungkin most probably it can help Dr. Paranita to answer her question.
Absolutely, I saw that question. You can definitely sequence across from paraffin embedded tissue. The problem comes from the crosslinks that are created from the DNA from the paraffin itself. So when you fix the material inside the paraffin, what happens is that results into these crosslinks in the DNA. And then when you sequence that, those crosslinks look like somatic mutations because of the way the enzymes work. And so what happens? Well, you can extract DNA from paraffin. It tends to be degraded, but that you can sequence from it. But the problem is you see a large number of artifacts. So what apparently looks like a lovely DNA sample, a DNA mutation, will look like a regular paraffin artifact. So it's very difficult to distinguish what is a true somatic mutation, the thing you're looking for, and an artifact from paraffin. Now, the interesting thing is that there are these kits now available that are able to repair that damage. Okay, now they're relatively expensive per sample, and that means you're able to repair the damage before you sequence it. And that dramatically increases the performance of next generation sequencing on paraffin. But you still probably would be unwise to do that on, say, example, whole genome or even whole exome. Whole exome starts to become complicated. It's possible, but it's just when you get the results back, it's very difficult to interpret them at the best of times and the best quality samples. But when with paraffin, it's even more complicated. So in the context of clinical practice, it's very complicated. You need always a validation step. If you find a mutation, well, we need to go off and independently validate it using an alternate technique to make sure it's real. In the research, it's then sorting what is real and what's not. Now, it's always better to have some sample than no sample. So if the only option is to get paraffin, so be it. And it's up to the lab folk to actually work on a better solution, because obviously for histolabs, paraffin is the method of the choice. It is, of course, so much better to look at the morphology of tissues when it's fixed in paraffin. So that's not going to change, and it's a matter of then working around. Ideally, collect paraffin and then take a small biopsy for next generation sequencing next to it and store that at -80. It does make all the labs work, and then the bioinformatics a lot easier.
Okay. Thank you, James. Jadi Mbak Vero, ya memang kesulitannya tinggi, tadi sudah dikatakan oleh Dr. Gunadi, kesulitannya tinggi, dan nanti kalau sudah ada hasilnya pun, itu nanti interpretasinya harus sangat hati-hati, apakah dia memang benar-benar ada variasi, atau variasinya itu dikarenakan tadi adanya crosslink karena kita memakai formalin. Baik, James, thank you for the sharing. So I will continue to the next question. Ini dari Mbak Like Nindita kepada Dr. Gunadi. Itu di Sarjito, siapakah yang melakukan pemeriksaan NGS? lab khusus genetika, lab penelitian, atau instalasi lab terpadu? Kemudian pertanyaannya kedua, bagaimana integrasi dengan sistem informasi laboratori? Apakah ada syarat fasilitas khusus untuk bioinformatik pemeriksaan NGS? Kemudian siapa yang bertanggung jawab atas hasil pemeriksaan lab NGS? Apakah SPPK atau spesialisasi lain? Monggo, Bu Nadi.
Terima kasih. Ini pertanyaan yang sudah kita diskusikan di tim Sarjito, bahwa nanti tentunya harapannya Pak Menteri, ya, ini sekedar sharing bahwa ini tidak hanya berbasis riset. Jadi Pak Menteri doesn't care about the paper, doesn't care about the research. Ini harapannya adalah pelayanan. Jadi harus mendukung pelayanan, sehingga proyek-proyek yang didukung oleh beliau itu memang akan menjadi satu untuk meningkatkan pelayanan di rumah sakit. Itu kenapa yang ditunjuk adalah rumah sakit vertikal, karena fokusnya adalah pelayanan. Pertama itu. Kemudian yang kedua, memang nanti kalau di Sarjito akan embedded ke dalam instalasi lab terpadu yang seperti disampaikan oleh Dr. Like ya. Like ya kalau tidak salah. Ya, Dr. Like. Dan sistem informasinya memang sedang diintegrasikan, Dok. Kita sedang berdiskusi dengan sistem informasi di Sarjito, dengan tentunya dengan DTO, tim Digital Technology Office-nya Kemkes, karena datanya tidak hanya di Sarjito, tetapi juga akan dilingkan di dalam DTO pusat. Jadi kan kalau dalam program BGS itu ada 3 ya, Biobank yang di-lead oleh Bu Yaya, kemudian Bioinformatics dan Bioregistry. Nah, kita bicara nanti Bioregistry itu tadi di-link dengan sistem informasi laboratori sana. Nah, kemudian untuk pemeriksaan nanti yang bertanggung jawab siapa, saat ini memang oleh tim genomik saat ini, dalam artian meskipun nanti di dalam lab instalasi lab terpadunya Sarjito, tetapi nanti kan di dalamnya misalnya pemeriksaan ini akan di sini, pemeriksaan ini di sini, begitu, Dok. Kan seperti PA, patologi anatomi kan juga di bawahnya instalasi lab terpadunya Sarjito, tetapi tetap yang bertanggung jawab adalah SPPA. Begitu, Dok. Yang sampai saat ini diskusinya terakhirnya seperti itu, jadi tetap yang bertanggung jawab untuk tim genomik seperti itu. Sama seperti pemeriksaan sitogenetik yang dilakukan di Sarjito, itu juga yang tanda tangan adalah SPA. Karena memang yang mempunyai kompetensi adalah hal ini adalah SPA. Meskipun mungkin masuk dalam rumahnya instalasi lab terpadu Sarjito. Mungkin itu, Dok, diskusi kami terakhir dengan tim Sarjito dan instalasi lab terpadu dengan direksi. Terima kasih pertanyaannya.
Terima kasih Dr. Gunadi atas jawabannya. Terima kasih juga untuk Dr. Like atas pertanyaannya. Ini tadi ada sedikit di chat dari Bu Dewi mengenai untuk CRC. Bu Dewi juga bekerja sama dengan Newcastle University menggunakan blok parafin untuk NGS. Memang hasilnya kurang baik, jelas ya, dibandingkan dengan fresh tissue, karena dari 10 sampel dari hasilnya DNA-nya yang cukup baik ternyata baru untuk NGS hanya separuhnya. Nah, tadi dikatakan oleh James, mungkin DNA-nya tampak cantik, tetapi harus tetap hati-hati untuk interpretasi datanya. Tadi jadi nanti untuk bioinformatics-nya tampaknya memang harus lebih berhati-hati lagi. Baik, mungkin ada pertanyaan lagi sebelum kita, masih ada waktu sekitar 7 menit. Mungkin ada Bapak Ibu yang mau bertanya langsung dengan open mic dan tidak menulis lewat chat box. Monggo dipersilahkan. Oke, mungkin sudah tidak ada, jadi mungkin saya ucapkan terima kasih sekali. Thank you so much to you, James dan Dr. Gunadi, that already give your valuable time for us to share your knowledge and your experience on next generation sequencing. I hope in the future we can have a nice collaboration between IARC and UGM. Dan kita dengar bahwa NGS itu memang adalah pendekatan yang baru, tapi tadi kita perlu juga mengingat apakah tergantung dari tujuan riset kita, apakah NGS ini bisa tidak selalu harus dilakukan. Jadi kita bisa melakukan pendekatan-pendekatan yang lain. Kami sebagai moderator akan menutup acara ini. Apabila ada yang kurang berkenan selama kami membawakan acara ini, kami mohon maaf sebesar-besarnya. Terima kasih banyak atas partisipasi dan kesabarannya mengikuti kuliah tamu ini. Saya lihat jumlahnya tidak berkurang sama sekali, malah tambah terus sampai semakin siang. Kita tutup acara ini dan kita ucapkan selamat melanjutkan kegiatan, especially for you, James, because there is still morning, so you have a lot things to do during the day. For us, it's almost the end of the day because it's already 3 o'clock here. So wish you luck. Saya ucapkan salam. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih.
Thank you very much, James.
Terima kasih. Thank you.
Terima kasih. Selamat siang.
Selamat siang. Good morning, James.
Selamat pagi.
Selamat pagi.